Namaste to all. Welcome to the next session on cell biology and genetics. So in this session, we are going to see about the mitosis cell division in eukaryotic cell. As we have already seen that how the mitosis is taking place in 4 stages in their karyokinesis, and in cytokinesis the division has already started and it has taken in form of 2 daughter cell. Let's compare and see that how it is happening in animal cell and plant cell. If you see in the prophase, before itself, the animal cell and the plant cell, if you compare, animal cell is going to have centrosome or the centrioles which are present. Plant cell, they are absent. So in the prophase stage, in animal cell, 2 asters are going to be formed, and in the late prophase, they are going to be present at the 2 poles, forming as amphiastral. And the spindle fibers starts to arise and they are going to join with the particular chromosome. In plant cells, since there is no centrioles present, so aster formation will not take place, but here there is a formation called as tubulin. So the tubulin undergoes the polymerization to form as spindle fibers, due to which this process is called as anastral. In metaphase, if you see, the chromosomes are getting aligned in the equatorial plane. The spindle fibers are getting attached to the kinetochore. Now, if you see in the plant cells, here also the same process is taking place. They are aligning in the equatorial plane and getting the tubulins which are formed or polymerized as spindle fibers are getting attached with the kinetochore. In the phase of anaphase, if you see, the cytokinesis starts at the end of the anaphase. But in plant cell, this will not happen in anaphase. In telophase, the assembly of all the organelles will start, and the disappearance or the dissociation or disassemble of aster rays will take place. Thereby, the spindle fibers and aster rays get dissociated and disappears. Cytokinesis continues, invagination happens, and it continues. If you see plant cell, this will begin only in the telophase, end of the telophase. So the last phase in this is the telophase. So in the telophase, the invagination increases, the division occurs and forms as 2 daughter cells, and this is like a centripetal form, which that is the invagination happens in the equator, move towards as a furrow, and thereby they joins towards inside. In plant cell, there is a formation called as cell plate formation. During the cell plate formation, if you see, there is a particular organelle or formation called as phragmoplast. The phragmoplast are the pieces of endoplasmic reticulum and the Golgi apparatus which comes in the center of the cell. During this, the vesicles of the phragmoplast releases some materials from this particular structure, and this is called as middle lamellae. This process is called as centrifugal manner process. Why? Because in this they move towards the periphery, and from the center they are going towards the other side, that is the cell wall side. So in this also there is no division of the cell is going to take place. So in the plant cell will not divide, but they will have a middle lamellae formation which is called as cell plate formation in the stages. But in animal cell, there are 2 cells which are formed which are called as daughter cells. Let's see about the types of mitosis. If you see the types of mitosis, the first type in this is anastral mitotic division, which we have already discussed in our previous slide, which is taking place without the aster formation, and this mainly occurs in plant cell. The second type in this is amphiastral mitotic division. Amphiastral, as we have seen during the phases, the asters or the centrioles are moving to the opposite side, aster rays are formed, so that is why these are called as amphiastral mitotic division. So here 2 aster rays are formed, and mainly we can see this in animal cell. The third in this is extranuclear mitotic division. Extranuclear mitotic division, you can find this in both plant cell and animal cell in the lower class or in the normal higher animals or higher plants. Intra-mitotic division. In this intranuclear mitotic division, we can find this is taking place inside the nucleus, and nuclear membrane is present as such, so it persists, and the spindle fibers are inside the nucleus only, and mainly we can find this in protozoans, some protozoans and some yeasts. Another type in this is called as endomitosis. As the term says, here the DNA duplicates, chromosome duplicates, and in this it results in polyploidies. That is, the chromosome will not split and the cell also will not split, thereby leading to different forms of the chromosomes, which we can see as polytene or lamprey. Mainly we can find in some of the liver cells and platelet cells where the endomitosis is present. Another one which we can see here is free nuclear division. Free nuclear division, in this the karyokinesis is not formed by cytokinesis, and it results in the formation of multinucleated cells, and mainly we can find this in fungal cells without the septa, or opalina, where the hyphae or the septas are intact. And multinucleated coinocytic or senescial, and dinomitosis, the last type in the mitosis. Here you can find this in the dinoflagellates, and nuclear membrane it persists, that is why this is called as mesokaryon. No spindle fibers are formed and chromosome remains attached to the nuclear membrane. So what controls the mitotic division? So when we talk about the control of the mitotic division, the first comes is the cell area to the volume ratio. So let's see. Normally, as per the mathematics, cell area we say this in square form, and the volume we say this in cubic. So if we take a cell and we make this as 1 centimeter by 1 centimeter area, so it becomes as 1 centimeter square. Volume as 1 centimeter by 1 centimeter by 1 centimeter, it becomes as 1 centimeter cube. So the ratio between the area and the volume is 1:1. The second type in this is the 2 centimeter by 2 centimeter, which is 4 centimeter square area, and volume if you see, if we take 2 centimeters, it is 2 into 2 into 2 centimeters, that is 8 centimeter cube. So if you take the ratio of this is 4:8, which is 1:2. So this is how this can be controlled. The second form in this is nucleocytoplasmic ratio. If you see the nucleocytoplasmic ratio, it depends on the nuclear mass and the cytoplasmic mass. So this is called as nucleoplasmic NP ratio or mass NP index. Mitogens. Mitogens, they are the triggering factors or triggering substances of mitosis. This can be hormones as well as certain environmental factors like cyclin, auxin, gibberellin, insulin, cytokinin, and temperature. Let's see about in this mitotic poisons. If you see, these are the chemicals or agents which are in the solid, liquid or gaseous forms which could inhibit the mitotic division during the process, and this is called as mitotic poison. So let's see some examples of this mitotic poison. The first in this is azides and cyanides. Many people know that when they take cyanides, they will die immediately. But what is happening in this? In this, the basic thing is they stop the mitotic division in the prophase itself. So this is like a chain reaction. So if in one cell this has occurred, throughout the body this will going to take place. So now, let's see the second in this is colichin. This comes from a particular plant, that is the Colchicum autumnale, and this stops the process during the metaphase. Since it is stopping the metaphase, now the chromosomes which has arranged themselves in the equatorial plane will not divide now. The second which is going to take place is the spindle fiber formation will not take place. And what has happened is we can obtain polyploids or chromosomes like which can take place in liver cells or platelets or in certain like polytene chromosomes form. The last in this is the mustard gas. Mustard gas, if you see, it causes the inflammation in lungs, oxidative stress, and leads to apoptosis of the cell. So most of the Nazi period, if you see in the World War II, during that time, the mustard gas was tested in humans as an inhuman practice during the concentration camps. The last in this process is carbon monoxide. Everyone knows when they are driving their vehicles, the exhaust which is coming up, in that carbon monoxide is present. Along with that, in industries also, when the chimney release is happening, from that the fumes which are coming up, that is also containing carbon monoxide. So let's see about the last in this, that is the carbon monoxide. In this, it stops the Krebs cycle. That is, mainly what happens during the Krebs cycle is, when they stop the ATP generation. Due to the stopping of the ATP generation, the chromosomes will not get the ATP, and indirectly they are going to stop the mitotic division. So this is how the mitosis gets stopped, and the persons who are inhaling carbon monoxide, they will have shortage of the cell division. And in the later stages, they can also have the wrinkles formation, seeing on their face old age, and they will become tired, or sometimes they can lead to brain death also. Let's see the next one in this, that is cancer. So this is an uncontrolled cell division which can happen at any place in the body. Substances which causes the cancer, we call them as carcinogen. And carcinogen, what they do is, when they are present, they trigger the cell for uncontrolled cell division, and the cell loses the control. Thereby, they lead to cancer. And once the carcinogens affects the gene, they can have many impact in the next generation also. So it can happen in somatic cells. Also, this can happen in vegetative cells also. Let's see the examples here. So benzaprine, so from the tobacco you can find this. So when the person is inhaling or particular tobacco is being smoked, then what happens is the active smoker as well as the passive smoker can have this lung cancer. The second in this is aflatoxins, which are released by Aspergillus, which is a fungus. And once it is being consumed by anyone, the food which is contaminated by this aflatoxin or like a canned food, from that we can find that that person is getting liver cancer. The third in this is vinyl chlorides. So PVC, which is a common example which everyone knows, that when they are constructing any house, the PVCs are used for their decorative purposes or for making the tables and other substances. So if any product which is kept for a longer period of time in PVC material, the chances of getting the liver cancer is higher here. So this is how we can understand that the mitotic divisions are very, very precise and in the equational manner. In the next session, we will be seeing about meiosis and how the division takes place during meiosis. Thank you. Have a great day.